everyone, my name is Shirin Parvin and I am from the Department of Electrical and Computer Engineering, Iowa State University, Iowa, United States. Due to visa issues, I couldn't be here to present my poster in person. But thanks to the IEEE Census 2025 conference organizers, I'm able to present my poster in the form of this presentation. My paper ID is 5607. I'm going to be presenting on in-tube sensors for continuous monitoring of cell cultures. Let's go in. Microbial fermentation offers sustainable routes for manufacturing of several high-value compounds such as pharmaceuticals, nutraceuticals, biofuels, and specialty materials by the usage of microorganisms such as bacteria, yeast, fungi. Real-time monitoring of these cell cultures by the usage of cell densities and dissolved oxygen is critical for optimizing the growth of these cells. Yet, even though these processes start in the lab scale shake flask cultures and small bioreactors, there is no integrated system for measurement of cell density and dissolved oxygen. For cell density monitoring, the current practices are time consuming, error prone, and can cause contamination risks, and also provides delayed feedback. To address these issues, we have developed a system which is compact and low cost and has the easy integration with a shake flask or small bioreactor. In our system, we have the dual sensing capabilities of fluorescence-based dissolved oxygen measurement as well as the backscattering-based cell density measurement. Now, let us go and look more deeply into our design of the in-tube sensors. Our experimental design is that in the shake flask, there will be the cell cultures, which will be pumped out using a pump and pass through our system. After processing through our system, it will be discarded as waste. For dissolved oxygen sensing, we use a dye called as PTOEP. This PTOEP dye exhibits fluorescence, which is quenched specifically by oxygen. For cell density measurement, we are using the backscattering of the light when cells pass through our system. Our system, as shown in this schematic, consists of a housing which houses the sensing tube. The inner walls of the sensing tube is actually dyed with the PTOEP dye. Then, other than the sensing tube, we also have the signal control PCB. This signal control PCB consists of two LEDs, one at 600 nanometers for measuring backscattering and another for excitation of the PTOP using a 390 nanometer LED. Both the signals, the fluorescence as well as the backscattering, is collected using the photodetector. Then this collected signal is amplified using the trans-impedance amplifying circuit, and then the signal is processed using an oscilloscope and a computer. Let's look more into our optical detector. This is the 3D rendered view of our designed system. Again, this is the housing, the sensing tube, the optical layer, and the trans-impedance amplifying circuit. Here, we have the real image of the sensor that we have fabricated. As we can see using this scale, that our system is actually very small and compact. For measurement of the dissolved oxygen, what parameter we take into consideration? We measure the fluorescence lifetime of the PTOEP dye. The PTOEP dye can be excited using a 390 nanometer UV LED. The emission from the PTOEP dye can be collected at 645 nanometer. And then this is collected by the photodiode to determine the fluorescence lifetime of the dye. As we know, because it is based on the principle of fluorescence quenching, the higher is the oxygen in the tube, less will be the fluorescence intensity signal. As the oxygen consumption occurs in by the cells, the dissolved oxygen in the tube is lesser, and then it results in the increase in fluorescence. For cell density measurements, we use a 600 nanometer LED as the light source. The cells, as it passes through the tubes, will scatter light back Then this is collected by the angle photodetector, which gives us the cell density measurement. In terms of signal processing, for the dissolved oxygen signal sensing, we input impulse signal at a time difference of 250 microseconds. During this time delay, we also measure the fluorescence decay that is exhibited by the PTOEP emission. For backscattering signal, we measure our sample, our signal systems at a frequency of 4 kilohertz. We convert this time domain signal into frequency domain using fast Fourier transform. Now, how do we calibrate our in-tube sensors? For calibration of the dissolved oxygen sensing part, we pass through gas mixtures containing various amounts of oxygen and argon through our sensing tube. For each of these combinations, we record the fluorescence decay results. To these fluorescence decay results, we fit a single exponential model to calculate the lifetimes. These calculated lifetimes are then converted into oxygen concentrations using the Stahl-Warmer equation as illustrated here. There, we found that the tau naught value is 20.1 microsecond and the Stahl-Warmer constant Ksv value is found to be 3.65. For cell density calibration, we grow a yeast cell called as Yerevia lipolytica over a time course of 28 hours. During this time, we measure the cell density in the shake flask using our system, and in parallel, we use the lab standard spectrophotometer based optical density measurement at 600 nanometers. We see that the signal that we measured using the backscattering using our system has a very strong correlation with that of the OD 600 measurements that from of a standard lab spectrophotometer. Further, to illustrate our system, we also culture two different kinds of yeast cells, Yerevia lipolytica, which is an aerobic cell, and Saccharomyces cerevisiae, which is a facultative anaerobe. That means it does not require oxygen for its survival. Using our system, we monitor over a course of 28 hours that Yerevia lipolytica consumes oxygen very rapidly and then results in a very low dissolved oxygen in the system for the remaining of the culture time. We also observe that Yerevia lipolytica shows a slow, smooth growing curve. Meanwhile, the Saccharomyces cerevisiae can be seen to be consuming oxygen at a much slower level, but it accumulates biomass very rapidly and grows very fast. This shows that the Saccharomyces cerevisiae does not need oxygen for itself to grow. This illustrates that our system can provide continuous and real-time tracking of both dissolved oxygen and cell density, demonstrating reliable performance across different microorganism profiles. In conclusion, we show that we have developed a low cost and very compact and easily integratable sensing system in which we measure both dissolved oxygen as well as cell culture densities. Using our system, we also illustrate that it can be utilized to distinguish between the different microbial growth profiles of different species-specific behaviors. This illustrates that our system has the potential for usage for a rapid and real-time monitoring of microbial cell cultures and bioprocess optimization. All this work was supported by the Bailey Research Center's Bailey Research Career Development Award to Iowa State University, as well as the United States National Science Foundation Award. I thank you all for stopping by my poster and listening to my presentation. Thank you.